Hello, everybody. My research topic for the ACE summer internship is tools to predict leaf nutrient levels. Talofa, good morning. Hola, everyone. My name is Chris Nye. I'm from American Samoa, attending American Samoa Community College, majoring in general agriculture. I'm an intern from Prosser. My research was done under the ACE Summer Research Program under the guide or direction of my mentor, Mrs. Bernadetta Salato. These are the contents. As we go along, I will do the introduction of my project, the objectives, method that I used, results, and conclusion at the end. Nutrient level prediction then guide adequate nutrient management, and it is very important to manage the nutrition part in any huge or big orchard because it can increase plant productivity, reduce the cost of chemical fertilizer, and the optimal application of nutrient at the right time is crucial for maximizing yield, minimizing nutrient losses. Leaf tissue analysis. So this is one of the most valuable and validated standardized tools to diagnose nutrients or monitor the nutritional status of any orchard. Limitation, as listed, so small representation of entire orchard, requires sampling, transporting, and laboratory testing. Chlorophyll, it does not relate to fruit quality, not useful for variability within an orchard. These are the objectives. First objective is compare tools for leaf nutrient assessment, and the second objective is correlate with three parameters, fruit size and shoot growth. Methods. So my project was initiated in an apple smart orchard in Mattawa, Washington, and the variety or the kind of apple was Cuspic Crisp. The sample were collected from 7 sites, 3 trees per site. These are the evaluations: standard leaf tissue sampling, HORIPA portable kit, SAP analysis, seasonal growth, and fruit count. The 7 locations chosen for these research are listed: Site 1, Site 2, Site 3, CC1 Site 1, CC1 Site 2, CC3 Site 4, and CC3 Site 5. In order to identify variability, the selection was informed by a number of data sources, including historical Google Earth satellite images, the current NDVI, these pictures sold down, and the ground data. Method. The first method is the standard leaf tissue method. I collected the leaf sample from the seasonal growth of an apple tree. As you can see the picture, this is the seasonal growth of an apple tree. And the maturely leaf, which are in between the young and the old leaf, non-bearing shoot, or non-bearing spur. So I collected the maturely leaf, which is in the middle. I circled the leaf. I collected 30 leaves per tree. Another method was the HORIPA portable kit. It used the exact same leaf that was gathered for the standard analysis. However, we squeezed the leaf and used this portable HORIPA nutrient kit to evaluate the trees rather than sending them to the lab. The third technique I will talk about is the SAP analysis, which was carried out as a third-party service by the Advanced Eco Ag Service, or the AEA. They sample on the days that we collected samples and provided producer with testing, recommendation for nutrient, and sampling. Results. So the site were relative homogeneous except for site 1 and CC1 site 2, as you can see on the data. So here in the graphs is site 1, CC1 site 2, and CC1 site 2. So first, our goal was to identify whether the data gathered within the orchard varied. Only Site 1 and CC1 Site 2 show a significant difference in shoot growth and Ricker indicated data. So continue on. Another crucial metric in the number of fruits, commonly known as the crop load. And it is evident from this that Site 1, the red one, differ only from Site 3 and 5. Site 3 and 5 in the yellow one. This result enabled us to pinpoint this variation and confirm that the range of value required to perform correlation between approaches is adequate. As you can see from this chart, there was no association found or relationship between the shoot growth and slender and leaf levels, and the same was true for all other nutrients. This is relevant information regarding the relationship between standard nutrient levels and shoot growth. This is a relationship between the standard nutrient level and the shoot growth, and you can see this chart, there is no relationship. In contrast, there was a negative correlation found between fruit nutrient level and Mg level. Mg level is not shown on this graph, and a significant relationship between potassium level is shown on this graph. So this is the relationship between the standard nutrient level and the fruit number. Standard versus key analysis. Only N indicated a significant negative correlation when comparing the HORIPA nutritional kit with conventional analysis. Additionally, there was a strong correlation between standard K and CA. In conclusion, there was a poor correlation between the result of the kit portable testing and the actual nutrient levels. We only found a positive correlation between fruit number and P and K in the olives, which is represented in the top chart. And spur leaf when comparing SAP analysis carried out by the AEA services without any further relationship. So the relationship between the SAP nutrient levels and the tree parameters. So these are the shoot, and this is the spur. Another way of looking at the correlation was by analyzing the differences by sites. The standard tests reveal variation between the site. This variation I show here in the N level, with site 1 and site 2 having the lowest level, and site 3 and 4 having the highest level in most cases in CC1 and CC3. These differences were also noted for the level of Mg and S. So according to the initial data, CC2 has the maximum vigor and site 1 in the lowest. So as you saw in this data, so the standard nutrient level different among sites. Standard B, K, and FE tests already reveal differences within the site, with site 1 having the highest level and site 3 and 5 having the lower level. As you may know, the fruit count differences between the site also indicated these variations. SAP level differences among sites. So in the same way, site 1 for B and K value were high compared to those of the site 3 and 5. That's why this is a positive or encouraging. In conclusion, HORIPA kit method did not correlate with nutrient level. Standard testing of K was a good indicator of fruit load and site differences. Standard N, while did not correlate strongly with shoot growth, it did reflect differences in deficiency area versus the adequate area site versus the other side. Sub-analysis of P and K also correlated with fruit number. Overall, the orchard nutrient levels were adequate to high except N level in site 1, indicated of deficiency, while CA level were low in all sites. So for my acknowledgement, I would like to express my deepest gratitude to my mentor, Mrs. Bernadita Salato, for her unwavering support, guiding, and invaluable insight throughout my internship. Your wisdom and encouragement has been an instrument in shaping my understanding and approach to this project. I am truly fortunate to have the opportunity to learn under your mentorship. I would also like to extend my heartfelt thanks to our team members, Juan, Uzi Reyes, Michael Oliver, Dayanara Gonzalez, and Brittany Sanchez. Thank you for helping me, and I've learned a lot from you guys. Thank you also to Miss Jordan Job and Miss Wendy Howe for your support and for putting this program together. This project would not have been possible without the collective effort, passion, and commitment of everyone who involved. I am sincerely grateful to all of you. Thank you so much, and God bless. Any question?